So, welcome everybody to this new season of our Memory Decoding Journal Club. As many of you know, we've been doing the Journal Club for pretty much all of 2025. Then we had an Aspirational Neuroscience event at the Society for Neuroscience annual meeting, where Ken handed out the awards for memory decoding related work. And we had some insights from that. So, based on that, we decided to get started again with the memory decoding journal clubs fairly quickly so that we can start the 25-26 season and hopefully do a few reviews before we dive back into brand new results. And today, I'm mainly covering the paper Learning Fast and Slow, Single and Many-Shot Learning in the Hippocampus. This is from the Losonczy lab, and I picked this paper for a specific reason. I picked it because what I did first is to try to get a lay of the land of what research in hippocampal system and episodic memory is currently like. And I wanted to get a general overview, kind of reviewing that research. And then I was hoping to pick a paper that intersects with or covers many of the bases in that field. Not everything. You'll see that there are some bits missing, and I'll point those out, but quite a few of them. And it's not a very long review paper, so that helps too. All right, so I'm going to tackle this in 3 steps. First, I'm going to say a few things about the reason why we're now doing a review paper, and we'll probably do a few more review papers in the next few sessions, and approaches that one could take. Then I'm going to dive into this paper, and I'll throw in a couple of bits from a separate, different review paper that Hasselmo and Stern did. That's Michael Hasselmo, who happens to be my previous mentor, because there are some bits there that the Losonczy review doesn't cover and that I think are relevant. And then afterwards, there's a piece where I take an approach that isn't directly related to the review paper to look at how we can dig through the hippocampal system and move towards memory decoding, figure out a memory decoding proposal. So first this bit, purpose and approaches. So here's what happened. The Aspirational Neuroscience event at the Society for Neuroscience was extremely well attended. There was clearly a lot of interest in the topic of memory decoding, and that was great. But we did find that there were repeated questions about what is a memory or a memory engram? What does it mean to decode a memory? What is a non-trivial memory, or what are non-trivial memories? How do you decide how many engrams you found and how many are enough to win the prize? All these sorts of questions came up over and over again. And that's not the first year where that happened. So it became clear that what we should be doing is we should be looking at a few different systems. Here are a couple of examples, or a few examples, like mouse hippocampus, zebrafish, the bird song, and Drosophila. And we could think about what would make a good memory decoding study or proposal in that model animal or animal model. And then write that up in a way where this isn't a prescription. It doesn't say you have to do one of these things, but here's an example of what we would find really compelling, something that would possibly win the prize. And well, on the bottom there, I've attached a little bit about what the Carbon Copies Foundation intends to do with that, because we work on in silico models where we generate something that is meant to produce a data set similar to what a researcher would obtain, especially when it's static data like electron microscopy. And what we would like to do then is to generate some examples that correspond with these proposed studies. So with that in mind, how do you start looking at the field of hippocampal research these days. And one way to go about it is to try to figure out who are currently the main experts, the leading principal investigators of the field, who have done very relevant and significantly progressive things that are moving on the field in recent times, say in the last 5 years, maybe a little bit longer. And this is definitely not a complete list, but this is a list that begins in a way with the principal investigators I was already familiar with back when I was working on the hippocampal system. And then forward from that, partly, uh, the new principal investigators are actually prior students of these previous investigators, and sometimes they come from another corner. And I just want to point out a few here. So Neil Burgess is very important in the field. Of obviously, I think that Attila Losonczy is making a name for himself. Otherwise, I wouldn't have picked this particular paper. Um, and then there's Michael Hasselmo, who then has two people, for instance, who've done a whole ton of new work: Mark Howard and Aaron Newman, who used to be in the Hasselmo lab. There's Matt Wilson, Edmund Rolls, Alessandro Treves. Edmund Rolls is actually not that involved anymore, at least not where Hippocampus is concerned. But Treves has still written some recent reviews. Um, these are these are obviously somewhat older researchers. They're again people who Michael Hasselmo studied under, for example. Then we can't forget about Buzaki, with all the dynamics and interneurons, and of course Howard Eichenbaum, who unfortunately isn't with us anymore. But one of his previous students, Steve Ramirez, is doing some great work. And then I also pulled out John Lissman and Ole Jensen, because they're not really focused directly or weren't focused directly on the hippocampal system. That wasn't their main thing, and it still isn't Ole's main thing. But some of the models that they came up with ended up being very informative and useful to hippocampal researchers. There are lots of different ways you could go about this. But okay, so these are, and then I found a whole ton of papers, of course. So there are, I don't know, 57 more that I want to go through to figure out what's really going on. I was only able to dig through some papers before doing this talk. So you could approach the whole lay of the land in a number of different ways. So, for instance, you could look at different temporal stages. You could look at acquisition stage, encoding, consolidation, retrieval, updating, even the gistification of memory. You could look at which kinds of retrieval are more or less dependent or independent of hippocampus after consolidation takes place. You could look at different types of memory or the dimensions of memory that are involved during hippocampal encoding. So, what sorts of information gets encoded by the hippocampus and along what dimensions? You could think about more mechanistic things like looking at place cells, grid cells, how grid cells become place cells or inform place cells, the new time cells notion that Howard Eichenbaum came up with before his passing, mechanistic evidence. So, how do the synapses change during encoding, during re encoding or reactivation? Is it more dendritic? Is it more somatic? Um, you could look at circuitry, architecture, and what that supports and constrains. Then you could look at modern data collection methods. Obviously, we've got, you know, electron microscopy that has produced very large maps in high detail, but now we have, other methods like Lycon that could be very useful, and uh, synaptic tagging and caging and all sorts of beautiful new techniques that could have an impact on the field. And then, of course, there's the models and theories approach, where the main interest would be how does all of this evidence come together, and how does that inform memory in the hippocampus, memory supported by the hippocampus, and then the decoding of memory that we're really interested in. Could go about it in all these different ways, and maybe we'll pick something in the future that makes the most sense as we're going towards a memory decoding proposal. But first, this paper. So the Liao and Losonczy review, and I'm putting in just this tiny bit from a different review paper by Hasselmo and Stern because of certain things that I want to include in there. Okay, so this review paper is mainly focused on trying to look at different kinds of learning in the hippocampus, namely single shot learning and many shot learning, where single shot learning means being able to acquire a memory from a single presentation of an event, and many shot learning means that you repeatedly explore an environment, repeatedly encounter sequences that are similar or even identical if it's a laboratory setting, and you're able to learn from that and develop cognitive maps. So let's have a little look at what kinds of cognitive function are dependent on the hippocampus or critically dependent. There's the mapping of environments and spatial navigation. You could extend that, and that has also been proposed in many papers. You can extend this to other types of conceptual relationships beyond the spatial. And I mean, we presume that in humans, for example, we use the hippocampus to relate a lot of different kinds of concepts, especially when we want to relate them in episodic memory. And episodic sequence memory or declarative memory is also very dependent on the hippocampus and particularly dependent. Now, there are different modes of operation in the hippocampus. So there's the so-called active or online operation. That's the type of operation where you see a lot of theta rhythm in the brain when the animal is moving around, exploring an environment, learning new things, exposed to new stimuli. And then there's the type of operation at rest, offline operation, where you see a different kind of activation in the hippocampus and in related areas like entorhinal cortex, typically not theta, but these sharp wave ripples. And there's a different mechanistic mechanism behind it and also different functional purpose in a sense, but we'll get to that. Then this paper in particular wants to distinguish clearly between learning modes and mechanisms depending on spike timing dependent potentiation or behavioral timescale potentiation, so BTSP. And they present some good reasons why you would want to have one or the other type of learning available at synapses. Now, important to remember is that for single shot learning, we have some requirements. You need to be able to acquire memory from a single presentation of an event or a sequence. And it needs to be reliable and persistent enough so that that novel memory can be used in the future, is available for retrieval. And many shot learning means we have repeated exposure. You can gradually mature a memory. And the memory doesn't necessarily get attributed to an individual experience, but you may end up with something that's more generalizable across different kinds of exposure and maybe even new environments. Okay, I guess it makes sense to take a look at the mouse hippocampus first, to just take an anatomical view and a functional view, take a look at it. And Ken, of course, pointed out the hippocampus book, which is a great place to go. So in the hippocampal system, the way it's drawn here, it looks a bit messy, a bit full. You could draw it in a different way, more as a, kind of a nicely rectangular system. But I thought it'd be nice to begin with this review just by looking at it within a brain in the context of the shape of the brain of the mouse brain and the mouse hippocampus and where things are. Just give it that appearance. So we have here on the left the hippocampus, on the right the entorhinal cortex. And so the entorhinal cortex has a number of different layers and regions. We've got the medial entorhinal cortex and the lateral entorhinal cortex. And these feed into the hippocampus in different ways. So you could say that there is what is often called a where stream, so information about space, and a what stream coming from the lateral medial lateral entorhinal cortex, not lateral medial. That makes no sense. And what we find here is this somewhat complex looking network of dentate gyrus, CA3 and CA1. And the entorhinal layer 2 feeds into the dentate gyrus. Then we've got dentate gyrus feeding into CA3. I won't talk about CA2 and CA4 and other areas here right now because that would just complicate things further. You can see that there are some fibers, the commissural fibers that are connecting between hemispheres. We've also got the associational fibers in CA3, recurrence between the CA3 pyramidal cells. Then the entorhinal cortex also leads directly to CA3, so some of those inputs are coming there without going through dentate gyrus. Then we have our important CA3 to CA1 Schaefer collaterals, and CA1 is also receiving information directly from entorhinal cortex layer 3. And this is very interesting because you can imagine how the information from entorhinal cortex layer 3 that is still cortical representation and not that sparsified representation that happens in CA3, which we'll get into in a moment. This can help to then recode and associate the sparse representations in the hippocampus back to cortical representations. So the CA1 is the output of hippocampus, and so you need some help changing those codes back into a representation that makes sense if you're pointing to cortical representations. And CA1 output goes out through subiculum and prefrontal cortex, goes to prefrontal cortex eventually. So we've got all these different connections in here. And the dentate gyrus in particular is special in the sense that it's very densely packed, sparsely active, so there are sparse representations. You see adult neurogenesis there, which is either absent or rare in other areas of the brain. So the dentate gyrus seems to be involved with orthogonalizing patterns, creating sparse patterns out of the not so sparse representations that you'd find in the cortex. So here we have that functional view. I've already talked about that quite a bit as I was going through the anatomical view. There's information coming from the world. That sensory input goes through cortical areas first. Then when it enters the hippocampus, gets discretized. So you're separating those patterns coming in from entorhinal cortex layer 2. In the hippocampus, CA3 is largely involved with the encoding of sequential information, so multiple patterns coming in over time. And then on the output side, you have to map back out to behavioral readout to drive behaviors, drive decision making, all that kind of beautiful stuff. Okay, so we have sensory input coming in from the cortex, being converted into what we might call meaningful concepts that could be represented by as little as single cells or small groups of cells in the hippocampus. We can call these cells place cells if we're dealing with spatial information. We can call them memory cells if we're dealing with other kinds of concepts. Depending on the context, depending on the use, there are other names as well, like schema cells. You can see some papers here that you might want to check out for that. And then you learn sequences of these concepts. And you learn that from either the spatial experience, the sequential experience. So stimuli that are coming in while you're exploring or while you're sitting there and something happens or you're learning. Or this happens during continued sampling of the patterns encountered before during resting, where patterns can reactivate. And this can cause activity in the hippocampus. And there can still be plasticity and therefore learning at that point. And then, of course, hippocampus eventually feeds back out to cortex. And in general, you can see different kinds of retrieval or replay in the hippocampus. There's the fast replay, the replay that's associated with these sharp wave ripples. Or you can see a replay that's taking place at theta frequency. Now, if you're in a mode where theta is present, so the active mode, the online mode, it can be a little bit difficult to distinguish between reactivations that are happening at theta or acquisition or sensory stimulation in the hippocampus happening at theta. But you see hippocampal place cells reactivating or activating at theta frequency, and the phase of theta is rather important there. But we'll get into that in a moment. So the hippocampus eventually feeds output into subiculum and off into other cortical areas. Some of that reaches the prefrontal cortex, and that guides behavior. Now, I know that place cells are something that we often talk about in the context of CA1 and maybe CA3, but you can actually find the equivalent of place cells or place cells, if you just want to say that, all over the hippocampus and all different subregions, though they look a bit different. They can be a bit more sparse or less sparse or have somewhat different appearance in CA1 to 4, dentate gyrus, subiculum or presubiculum. But really, they happen everywhere. And that's not surprising because all these areas are connected and they feed into each other. Another important point is that the hippocampal interneuron populations tend to be highly synchronized. So it's, it appears that there are some interneurons in the hippocampus that may be more specialized for specific stimuli. But overall, in large part, interneuron populations seem to fire together. And that means that they are not as easily directly usable for coding specific concepts or helping to distinguish or encode different concepts, but they can be very useful for the suppression of activity in hippocampus, separating patterns, allowing sequences to be robust, stable, not as noisy. Now, I wanted to put in a quick aside, which was not in this review paper, because the review paper doesn't really address grid cells, but I'm sure some people are curious about those because grid cells have become rather famous. These are cells in the entorhinal cortex that fire in a way that is very patterned, very repetitive, hexagonal patterns at different scales, fine patterns of hexagons and coarse patterns of hexagons. I have a couple of examples here that are from either models or in vivo data that are from the Hasselmo and Stern paper, just so that you can get an overview of what that looks like. So everywhere where there's a dot, that's where one of those grid cells is firing. It's the same grid cell firing in multiple locations, very regular patterned way. So what's happening here? The hippocampal place fields, they receive this grid cell input. The grid cells are driven by the sensory information that's been processed through the cortex. And then the hippocampal place cells receive grid cell input from medial entorhinal cortex. But they also integrate directional cues. So it's not purely grid cell information. Place cells in the hippocampus can also be head direction sensitive or sensitive to other information like boundary information. Then a Hebbian learning rule can strengthen the connections where grid cells fire, and that's happening at the same time with the sensory input. So what you end up with is that you can use multiple grid cells to provide different vectors that you then combine to create a stable place cell or stable place cell activity. And so then you end up having a cell that associates with one specific location instead of an entire grid. So in the review paper, Losonczy also mentions that this notion of the non-local replay, so the ability to replay sequences offline when you're not in a particular location or online when you're planning what to do, is very useful to support decision making. And this is now also being used in some AI models. For example, there's this Demis Hassabis paper here that includes a number of different bits and pieces of neuro-inspired artificial intelligence. And one of the things that they take on board is how useful sequence replay is in reinforcement learning. But an important thing to keep in mind is that replay doesn't mean identical replay of sequences, or it doesn't have to. It can, but it doesn't have to. You end up with a replay that contains either unobserved activations or activations that correlate with unobserved stimuli, or that produce sequences that might be impossible in the learning environment. Some salient features might be omitted, and the distribution of stimuli can be very different from observed, or somewhat different, not too different. Otherwise, you might not be talking about a sequence that you've previously learned. And yeah, I guess the last bit about this computational study part is that in models, you can show how the activity from one region builds up from activity in previous regions. There are a number of different models for that, and leads to generally the separation of patterns in dentate gyrus, the completion of patterns in CA3, either autoassociative, heteroassociative, i.e. sequences or both, and then complex composition at CA1 as you prepare to bring the output back out. Okay, now we're getting to the real meat of the paper, because first let's tackle single shot learning. Now, it's been known for a long time that the hippocampus is involved with and necessary for single shot sequence learning. There's a paper here from 1958 by Penfield and Milner, super famous. So as you explore a novel space, you find that place fields cause selective firing or bursts in cells in the hippocampus. And then you could come up with multiple different ideas or models of how that can lead to single shot learning. I took the liberty of putting one of my own papers in here as well. That's a model where buffering of sequences, which is something that you can find in medial entorhinal cortex and other cortical areas. It's a buffer that's based on oscillatory theta, gamma, and on after depolarization. That's one way of achieving single shot learning. Now, the single shot learning that they're more interested in here is the use of behavioral timescale plasticity. So you can get immediate emergence of place fields from BTSP happening in hippocampal areas. And while this is happening, this is all achieving compression of input over time. And it's constrained by sparsity. These are very useful, powerful things for the coding, the encoding, and the use of the information. Now, what you need is you need strong LTP and LTD, so both strong potentiation and depotentiation happening from a single experience. Spike timing dependent potentiation isn't very good at that, or can't do that, because every time that you have spike timing dependent potentiation, you get a very small change of the synapse, every stimulus. And you can imagine that this is also very sensitive to noise. So if you have noisy input, you don't get the same activity every time you're presenting a set of stimuli. You want it to be somewhat generalizable, not overfitting. So being able to average over a longer time scale of stimuli, a longer time scale of activation in the hippocampus, is something that can help guide towards reliable weight change. So behavioral timescale plasticity, that's promising for this, for novel acquisition because of that, because it averages over a longer period of time. And we'll see more about that in the next slide. The pyramidal dendrites receive a variety of tuned inputs Those can be either produced by endogenous signals, or you can generate them optogenetically. And then you see plateau potentials on distal apical dendrites. So these are the dendrites. If you know, a pyramidal neuron has apical dendrites and basal dendrites. They're on opposite sides of the pyramidal neuron. Apical dendrites tend to stretch out over long distances, and therefore you can connect with other regions through those apical dendrites. Basal dendrites are closer to the cell body and serve a different purpose. So you have potentiation of weights happening before and after signals, and the potentiation also happens on a large number of synapses. This makes it very robust. So BTSP is then expressed mainly in novel environments. That's when you see these bursts of firing happening, when there is this plateau potential on the distal apical dendrites, and happens over a large population. So you get large population activity, and both in CA1 and CA3. Here is the figure 2 from the paper that describes how this happens. So let's take a look at A here, figure 1A. This is showing us the two dendrites, or two different dendrites of your dendritic tree. Soma, input coming in from different sources, different stimuli on one dendritic branch, other stimuli on another dendritic branch. And here we have in B, we see this over time. We see the different inputs of different stimuli coming in. And we see that as this dendritic plateau potential occurs, we have strengthening LTP for the signals that occur before, the signals that occur after, so over this entire region. So here we have and see our strong BTSP event, a burst happening and taking place over several seconds, perhaps as much as that. So this is able to integrate over a number of different spikes, give you much stronger potentiation on the synapses. And then here we're seeing this expressed over time, over different laps, different trials, as an animal is running. And we can see that at first, there is no place field activity here. But then as the plateau happens and there's BTSP, the place field starts to form. We see that it begins to extend to previous and following positions. So it stretches out over larger length of the animal's run on its lap, and carries on like that throughout the experiment. And this is just showing us the change in what you would see in the membrane potential there at different laps. So lap 10 is before BTSP has occurred. Then lap 11 is when you get that plateau. And this can happen due to a multitude of inputs spontaneously in an area where the dendrites are already relatively well connected with those stimuli, and then strengthens. And then we see that as it goes on up to lap 27, you find this strong activation. And if you look carefully, you compare the size here of the 1 second, and you look at how rapidly these activations are occurring. This definitely happens over multiple theta cycles. Yeah, E and F, I won't stay looking at those for too long. This is looking at the correlation of the tuning curves between apical and basal dendrites, different branches on those dendrites. And here we have the sort of the distance between the somatic place field and the dendritic place field on apical versus basal dendrites, where you can see that it's fairly evenly spaced out here, and the basal dendrite definitely has a preference for a place field that's very close to the somatic place field. So the basal dendrite doesn't seem to be exhibiting that same ability to create these connections in one shot, for the one-shot learning between, say, one place and another place that you get from the apical dendrites. So a little bit about the dendritic substrate of single-shot learning. There's evidence that dendritic compartmentalization is involved in the process of creating selective cortical features, and so then you end up with these tuned dendritic spikes that are predictive of somatic tuning. But there is a dependence on brain state. So in some states you'll find this soma dendrite coupling, and in other states you won't. Yes, go ahead, Ken.
Sorry, I hope it's okay to ask questions.
Absolutely. Yeah.
So I was confused by this part of the paper when it was saying that it's essentially saying, okay, there's dendritic compartmentalization, so you have different feature selectivity of different branches. It's my assumption. Correct me. I'm wrong somewhere here.
That's
And
exactly
then it
right.
says that the local dendritic spikes are predictive of somatic tuning, which almost seems at odds with the previous statement. What does that mean that it's predictive of somatic tuning?
So the way I interpreted it is that once you've got dendrites that are more likely to fire for specific input stimuli, that you can predict how this will end up changing the place field selectivity and the connection with other place cells in that soma. So that it's predictive of how the cell will respond to input stimuli from, say, for example, a preceding sequence that you have, say, A, B, and then C, and not A, D, and then C.
Okay. I mean, if it was totally predictive, then essentially that would mean that it's not compartmentalized. So this is kind of somewhat predictive, but
this is the tuned dendritic spiking, right? So this is post BTSP.
yes. So help me understand how I'm supposed to be thinking about this. So I've got a soma response that might like a particular place field, and that soma response is clearly coming from a sum of inputs from all the dendritic compartments, but those dendritic compartments have their own place fields, or they have place fields that are less specific, but the soma is kind of averaging them all out.
Now, I think this depends on the difference between the apical and basal dendrites. That you've got the correspondence between the basal dendritic preference and the somatic preference, but that the apical dendrites are experiencing BTSP and are helping you establish in one shot, because of BTSP, these connections back through sequences that can be made up of multiple previous and perhaps some following activations, so that you have a path, a little sequence. So then after BTSP on those apical dendrites, the dendritic tuning in those compartments, those different branches of the dendrites, those become predictive of which sequences the soma will fire in. That's how I read it anyway.
Okay.
Does that make more sense?
I think so. I think so.
I hope I'm getting it right. I mean, the paper is a bit sparse information-wise in that spot.
It's a review paper. I didn't go and read the.
Yeah, me neither. I didn't go to all of those. As I said, there are so many papers still to do. Yeah. But that's how I was reading it anyway. All right. Okay, so now we switch from single-shot learning to many-shot incremental sequence learning and replay. So as we know, the place cells fire in predictable sequences as the animal explores. We're just putting together some information on the left here. An animal running through an environment, no matter how many different places there are, no matter how many dimensions you're dealing with, whether it's spatial or other concepts, if you have a sequence where there's one space, one location, or one concept happening after another, or something you can map to a concept happening after another, you always have a 1D trajectory. So you always end up with something that you can express as a sequence. We know that the 4 to 8 hertz theta dominates during exploration. So this is involved somehow. And we also know from some studies of, many studies of electrophysiology in hippocampal cells, that the place cell firing during exploration happens preferentially in one phase of theta, not in all phases of theta. And we also know that as the animal moves from location to location, the phase at which a particular place cell fires tends to shift to earlier phases of the theta cycle. So these phases of place cell firing are sorted by position or sorted by the order of some episodic event. And the sequence of firing in these place cells, in hippocampal place cells, has been shown to be predictive of a destination or a goal, at least in these laboratory conditions, right? We also see that at rest, the animal will show completely different place cell firing. We see this replay that is time compressed, sharp wave ripples. And this is happening when there's no theta present or very little theta. And it has also been shown that sharp wave ripples are critical for memory consolidation. If you suppress that, if you chemically suppress it, for example, or lesion, then this definitely affects the ability to consolidate hippocampal memory. And sharp wave ripples, even if the exploration of the environment happened in one particular order, the sharp wave ripples aren't always observed in forward order. They're also observed in the reverse order of the sequence, but still in sequential order, just reversed. That's interesting. All right, so this is where we get to take a little look at online and offline hippocampal sequences. It's figure 3 of the review paper. And it's showing a particular experiment or the runs of an animal in a maze, let's just say. And what we have in A is that we're seeing that as the animal is running, you get different place cells that activate in different locations. In B, we can see these same place cells happening, these place cells firing on different phases of the theta rhythm, and that they preferentially happen in one part of the theta, one half of the theta cycle. It's not exactly a half, something like that. And then conversely, in the offline mode, you've got these sharp ripples happening, some of them forward, some of them backwards. And then the idea is that you have Hebbian plasticity during exploration. This is happening during the online mode. This is strengthening the connections between different place cells due to strong cortical input. And then during replay in sharp wave ripples, you have some input, of course, that sets off firing in the hippocampus. There's also spontaneous firing, but there's weaker hip cortical input. There's no learning going on or very little learning going on, even though there is more about that in a moment, reactivation. So you get the sharp wave ripple retrieval. And in this paper, so they talk mainly about this happening in entirely different modes, so offline and online. But there is a different way of looking at the electrophysiology when recording from hippocampus, where it seems that sharp wave ripples can also occur in the opposing phase of theta during online modes. And this is something that, so the Hasselmo lab is very well known for studying that. I studied it myself as well. And this is something that for some reason isn't really included in this review paper. There's a tiny little reference to it, but not much. So I'm going to add in a slide here to talk about that a little bit in just a moment. Anyways, carrying on here first. So here we have our laps being done by the animal, where it's yellow, the animal is running, green, it's immobile. We see the positions, the cells that represent a position. So here the positions are being recreated from the cell activity. Got CA1 cells firing. And here's the theta band, and you see that they're preferentially firing at theta frequency. And in the fourth sort of sub image here, we're seeing a close-up here of the cell firing sorted by position. So that's why you get this beautiful line. This is, of course, not what you see if you randomly record in the hippocampus, but they're arranged by peak firing location. And then we have here examples of activity during resting, both forward and reverse sharp wave replay. So now I'm going to throw in this other model. And the reason I'm going to do that is because there are a few things, a few particularities that don't get mentioned. This is from the so-called SPEAR model, separate phases of encoding and retrieval, that the Hasselmo lab and labs associated with them have been working on for a while, although maybe not as much right now as in the past. And so this is again based on this evidence that, from electrophysiology, that's phase coding. So spike timing is relative to theta, that phases shift to earlier times in theta, which makes the spikes, the sequences of place cells that appear to fire on that particular flank of theta feel like a first in, first out kind of buffer, where eventually firing will drop away for one cell and then a new one comes in and they stay in that sequence, but they keep shifting to earlier phases in theta. And you find this same kind of phase dependent firing also in the grid cells. So this happens from medial entorhinal cortex all the way through hippocampus. And you also see that if you measure theta, extracellular theta in the field potential, that you find that there's a phase offset between regions. And it's a very interesting offset, because when you consider the flank of theta at which firing is happening, then it happens to align beautifully in the sense that, for instance, the cortical theta will happen at a phase where firing occurs just as the hippocampus is in a phase, so that anticipates encoding, that receives encoding information from cortex. And then when retrieval is happening in hippocampus, that happens at a time when the cortex is ready for acquisition and encoding. There's a lot more behind this model, so I'm not giving it its due by describing it like this. We should do a separate session on that. But what you end up with then is that you can, in a single theta cycle, find room for encoding information on one flank of theta, and in the other flank, there's enough room for a quick, sharp ripple. So you can have retrieval as well. And this can go on concurrently on the same cells, same populations, throughout encoding and retrieval phases happening during online active learning modes. And there's various support for each part of this if you go to the papers that I've listed here. In particular, there's a specific bit of evidence about GABA B receptors and their modulation at different phases of theta, and how this changes which connections are strongly processing and sending activation and which ones are weaker. And this is how you get a change between where recurrent activity in CA3 is weak during encoding, but strong during retrieval. There is strong LTP during encoding, no LTP during retrieval. But again, this is just one way of looking at it, and I think there's probably some contention between labs that focus on this particular model, or labs that focus on models where online and offline are completely separated. A bit more about replay. Now we're back to our review paper. So there are several roles for replay. There is its involvement in prediction or planning. So let's say you're trying to find a goal. It's goal-directed navigation, let's just say. And there's a role in storing or organizing. So changing the connections with, or the generalizability of the information you've already stored. Specific experiences are typically not replayed when you're in a natural non-lab environment. So this is, if you go to this paper, you'll find that that's relatively new evidence showing that the sequences learned in hippocampus under normal conditions are not typically retrieved as precise, identical replays of previously encountered sequences, but tend to be something more generalized or generalizable. The previous work that showed very specific replay of identical sequences, I mean, it's not wrong. This can happen, but it's not typical in the sense that this was done in lab environments where the rat only had a very constrained set of different sequences it could experience. There was a very constrained maze or set of stimuli while running, and that affects what the results are, of course. Now, filtering and selection is done in a way that emphasizes sequences that are more important to remember and replay. So and this is done, or at least the evidence shows that remote experiences learned in the past, are replayed more often during replay or offline modes, or at rest, or while preparing to start heading towards a goal, than immediate upcoming paths. Exploration with happens with random stimuli, but the replay includes mostly predictable stimulus sequences. So if you have random stimulation in your laboratory experiment, let's say you have some distractors, you'll find that the replay typically contains the more predictable stimuli and not the distractors. So what we have here is something that has reduced recency bias and focuses on the salient and not irrelevant stimuli, but how can that happen? So the idea here is that this replay has to transition to a more generalizable form of the memory as the animal is exposed to multiple instances of sequences, and this is learned gradually. CA3 has many recurrence. CA1 has few excitatory connections, so there isn't as much opportunity there for direct sequence learning. Sequences are learned in CA3, read out in CA1. Now, the place cells and sequences are in part determined by the pre-existing connectivity. So I don't get into this at all. There's a whole part in the paper about development, but I found that it wasn't super compelling and not as important for our focus on memory decoding. So I'm not going to get into that, but it seems that the prior existing connectivity from development may be largely involved in picking which cells activate as you're exposed to stimuli. And that makes sense, of course. So if you're going to set up sparse conceptual representations, yeah, that makes sense. All right, then there's a bit about the utility of inhibitory plasticity in Hebbian sequence learning. Imagine a situation where your rat is being exposed to stimuli as it's running on a treadmill. There are some random cues that happen at random intervals. And we're seeing that here. Then in B, there's the average sharp wave ripple response at the Schaefer collateral, so between CA3 and CA1 axons, or between CA3 and CA1 on the axons. Here we can see this shown on those axons with a zoom into the time component for the time before the the random cue, the time during the random cue, the random stimulus, and the time after the random stimulus. And what you see is that the place cells that are tuned to or selected for that random stimulus, they are suppressed during and after presentation of the random stimulus. And that's interesting. So how is that possible? Let's imagine here that we have a world in which concepts occur in some order, and you can have a number of these sequences where you have the concepts you're trying to learn. So the important, the relevant stimuli, the salient ones, blue, red, yellow, green. But then every once in a while, we have an orange in there, and this orange happens at different times, different points in the sequence of our random cues, which I guess aren't quite random here. Then Hebbian learning sets up a matrix of strong connections, which corresponds with the sequence that we have here. And that would include the spurious associations. But because this is not the same, I mean, this spurious representation is occurring at different points in the sequence. It's kind of random, right? If you set up inhibitory plasticity as well, then that inhibitory plasticity can prevent the replay of these spurious distractors while you're replaying your sequence. So here's a use for, at least, inhibitory plasticity, which I also didn't get into in greater detail because it seems like the evidence for it and where exactly it occurs, which inhibitory neurons are involved and which ones are not, isn't quite clear yet. But they're presenting this as another way to end up with more generalizable sequences instead of the specific sequence that you observed, right? Say, for instance, blue, red, yellow, orange, green. You've observed all these specific sequences, but what you end up with, what you can then retrieve and replay, are the more generalizable sequences. So the sequences you retrieve when replay happens aren't exactly the same as what happened during exposure, but they're in a sense more useful because they avoid replaying distractors and instead replay the most generalizable parts of the sequence. Now we have a set of rules for many-shot sequence learning, and also a reason why this can't really use BTSP. So BTSP, first of all, can't distinguish the order of stimuli within its interval window. It's just a burst that's happening over a long period of time, a second or more. And the stimuli, of course, in addition to not being able to distinguish them within that period, stimuli can appear at any interval separation and sequence length. So you could have a stimulus happening now, another one 5 seconds from now, another one a minute from now, or they could be happening much more rapidly. But again, BTSP can't distinguish the order within a small interval. And the length of that sequence could be anything. Could be 4 stimuli, 10, or 15. So BTSP, this single shot learning, can't cover how to learn these sequences in the way that many shot learning can. But STDP is useful in this context. It can handle having many different pairings in many instances of exposure to stimuli. And by being exposed to many different instances of those stimuli, even with noise present, even with slight differences in the exposure, by only making very gradual, tiny changes, you can suppress the spurious distractors and keep the more general salient information. So this is very well suited for learning on recurrent weights. You can bridge longer intervals in a number of different ways. And here, I think I mentioned a couple of those. So one of these is that shortly before Eichenbaum passed, he was well known for having discovered yet another kind of cell in the hippocampus. So in addition to place cells, grid cells, he discovered something that he termed time cells. These are cells that respond to different intervals of stimuli, so different intervals between stimuli. And again, you have, just like in grid cells, where you have these different hexagonal maps at different scales, you have time cells at different scales. I don't know exactly why time cells occur, but this is just a finding from electrophysiology. Then another way to bridge longer intervals, besides using these time cells to tie things together, or different intervals of time cells, is embedding in theta cycles that allows you to compress sequences. I was talking earlier about how spiking happens in a certain flank, a certain part of the theta rhythm, and that phase precession occurs where the phase moves forward to earlier phases of theta. And what you can see is that as new spiking enters the hippocampus, and you see a reactivation of that sequence where you see it slowly shifting to earlier phases of theta, newer place cell firing not only enters on that flank of theta, but it tends to shift closer to the earlier pattern, the earlier spikes, and then the next one and the next one. They can become time compressed in a sense, that you have stimuli that appear at maybe several hundred milliseconds separated, but then the reactivation of that in the hippocampus appears at the kind of separation that still allows STDP. So where you have the firing of different place cells happening within about 20 milliseconds. And there's, there are again models that show how you might achieve that. And one of those models is a model dependent on buffering at theta cycles, in theta cycles, buffering a number of different patterns separated by inhibition of interneurons. And I've put one reference here that is a paper that I wrote with Michael Hasselmo. But there are other models as well. Yeah, I can't get into all the different models here, obviously, in one discussion. The forward and reverse sequences that you see in the retrieval, there's a question about how you can achieve those forward and reverse sequences. And there are a number of different papers that show how symmetric STDP could be used to achieve both forward and reverse sequences. So instead of having the typical asymmetric shape that you see in the and Poo paper, you find that symmetric learning can achieve, of course, sequences in both different directions. A different and alternative way of doing this is, again, to use these short-term buffers that I was talking about before, where the timing at which input enters the buffer can determine whether the buffer is learning a forward or a reverse sequence. So there are multiple ways that you could achieve that. Oh, and this is just, I'm going to skip that. That was just a look at one of those. All right, so consolidation and reorganization. First of all, sharp wave ripples are obviously a biomarker for consolidation. They're required for it, and you also see them during consolidation in hippocampus and in cortex. There are complementary representations of activity in hippocampus and in the cortex. This is suggestive that there's a transfer happening as the performance improves over time with these complementary representations happening. And the replay is clearly coordinated between hippocampus and cortex. And consolidation is selective somehow. selects some sequences and not others. Not all memories are retained. Pattern reactivation in sharp wave ripples at rest induces plasticity. Disruptions of that sharp wave ripple activity impairs learning. And depolarization during sharp wave ripples enables synaptic plasticity on subsequent sharp wave ripples. There's strengthening of coactive CA1 assemblies and homeostatic depression of unrelated CA1 activity. Now, this reactivated representations, these change over time by amplifying the signal, and in doing so, they amplify the signal, they attenuate noise, and they transfer to cortex. Okay, we're getting to the end of this, and then I just have that onward bit afterwards. So summarizing over the paper, and I know some people have already had to leave. Sorry about my speed at this. Summarizing, behavioral timescale synaptic plasticity allows the acquisition of new concepts from single experiences. Hebbian plasticity supports the incremental assimilation of sequential information over many trials. There's a trade-off between speed and weight change that may underpin both BTSP and spike timing dependent plasticity, and replay sequences exhibits distinct statistics and content compared to sequences observed online. And then the paper mentions a few open questions that remain, such as what principles govern replay and its plasticity? Is there BTSP in other brain areas? And I think we've already found a few papers in previous journal clubs that may point to that. What are the expressivity and limits of hippocampal learning? Further investigations of inhibitory and offline plasticity are necessary. I already mentioned that, especially that inhibitory element is not very clear yet. Okay, so having done that, I think I'm going to jump over many of these things because we've already touched on them before for the review paper. I just wanted to point out that instead of looking at this the way that the review paper does, we can, for instance, look at this in terms of different topics, like the topic of acquisition and encoding, and what's going on there. What is the mechanistic evidence? If you're really interested in this, we can go back to these slides, but I don't really want to do that right now. Instead, I'll share the slides so you can read these pieces. We could look at early synaptic stabilization, so what's going on between minutes and hours. How does synaptic tagging and capture play a role in that? What is the evidence? We could look at systems consolidation, so this is a totally different temporal phase of what's going on. And there are two different complementary frameworks here. There's the consolidation model and a trace transformation type of model. Again, I won't dig into the details here, but so we see there are a number of different models still. It's not as if hippocampal research has coalesced around a single model at this time. We could look at retrieval, how you get the pattern completion and indexing pointers, basically, and how you pull the information out of hippocampus back into meaningful patterns in cortex. And then there's the whole matter of updating and modification, which maybe isn't as important for a memory decoding challenge, but it's super interesting, especially since there's been so much new work on what exactly is happening at synapses during reactivation of cortical information and how this then changes representations in hippocampus, that there's a drift, a change of patterns, and what this all means. So just summarizing that, rapid encoding stores an episode using separation mechanisms so memories don't catastrophically interfere. During online theta states, the circuit alternates phases optimized for encoding new inputs versus retrieving previous patterns. Early molecular consolidation decides which synapses will stabilize into long-term changes. Offline replay during rest or sleep drives hippocampal to cortical communication and redistribution of memory traces. And reactivation windows permit reconsolidation and updating, prediction errors, and neuromodulators bias what's consolidated or changed. And there's a list of particularly influential publications, but as I mentioned, there's a whole stack of new papers that we should still take a look at. I wanted to quickly point out that there's some good books we can use to get a better idea of the sort of the landscape of hippocampal research at this time and what things are fairly well understood and where there's a lot of consensus versus areas where there's still new ideas cropping up and models that are quite different from one another. There's the Hippocampus book by Anderson, Space, Time, and Memory by Lynn Nadell, which sounds like a really awesome book, and I haven't read it yet. This one here, the Oxford Handbook of Human Memory, has the benefit of being available online, and it's also quite useful for modelers to take a look at that one. How to change a memory, so that's kind of particular. This is how do you modify uh, through techniques? How can you modify memory? And then here, hippocampal microcircuits, this one specifically for computational modelers. I think that's all I have there. And then my very, very last slide to those of us who are still here is to try to suggest what we can do next. Because as you saw, I mean, there's a lot of material there. And because I had so many things to dig through, you could tell that I didn't have a lot of time to actually practice presenting it well. I'm very sorry about that. But here's some things we could do to move towards having a decoding proposal. So one thing we can do is that we can switch to looking at papers that explicitly link in vivo evidence, theoretical models, and static data, because static data is, of course, what we want to focus on for memory decoding study. And then the other thing, the next step would be pick an experimentally feasible set of regions or subregions. Some techniques could handle potentially looking at entire regions and their interaction. And maybe there are techniques that provide static data at that size in something like mouse. That would be pretty cool. But that may be way too ambitious. And it might be better to consider a memory decoding study where you look at a few subregions or one subregion, even just CA3 or CA1, or both CA3 and CA1. And then go ahead and define what we mean by here's a memory engram. This is what we consider a memory. What does it mean to decode one memory, two memories, three different memories? How many can we separate there? And at what point is that non-trivial? These would be the essentials, I think, once you get down to looking at a particular circuit. And then, yes, of course, write up a proposed study. So, opening this up to discussion. Yeah, go ahead, Ken.
Thanks, Randall. Let me say what I'm most interested in going forward is bringing very simple models that can say, here is a functional sequence, here is how a single theory says it would be encoded in the synapses. And let me throw one of these out there just to make sure if I'm totally off track, because I have not looked into hippocampal models except for a little bit of roles stuff, and not any sequences or anything. So I am assuming that this this theta cycle progression can be thought of as I have CA3. I have a set of ensembles in CA3 that are, let's say, ensemble A is mutually excitatory to itself, and ensemble B is mutually excitatory to itself, and ensemble C is such. But A is somewhat exciting B, and B is somewhat exciting C, et cetera, et cetera. And each of these gets input from the environment, from I don't know, entorhinal cortex, let's say. So I can bias those up as place fields, let's say. And then the theta wave comes through. And the theta wave is kind of saying, okay, whoever is most likely to fire, go ahead and fire. And A goes first. because it's getting the most input, but then its activation ripples through to B, ripples through to C, and that's where a sequence is happening. So help me out. Is that a viable model? Is that a model from 50 years ago that's been completely
No, that seems viable. I mean, everything that I've reviewed so far hasn't discarded that model. I think just looking at CA3 recurrence and focusing mainly on those makes a lot of sense. And I mean, as far as the retrieval reactivation data being enough, okay. If we assume that there's some noise in there and some spontaneous activity, something's going to reactivate at high theta. But you could also, say, okay, let's pretend that we're doing optogenetic stimulation of a few Q cells of a particular engram, and then see if the sequence comes back at during a theta cycle, for instance. That's something you could do, or even without theta, just sharp wave ripples.
Okay.
You could do either one. But the other thing is that doesn't seem. So you know how we were talking about the difference between our brain emulation challenge and the memory decoding challenge? And the brain emulation challenge would include showing the decoding happening. So having something like what you just described happening in there. But a memory decoding challenge, in theory, wouldn't even need to do that, right? Because you wouldn't need to show it actually doing the retrieval. You would just need to be able to identify the different ensembles.
Yes, although I suspect it would be, given that any experiment has a whole bunch of noise in it, it would be much better if there was calcium imaging beforehand that showed these sequences, and then
Sure.
you can verify that it had the connectivity that your model predicts.
Okay. So then
So
it's, this is the input information rather than the output of the challenge then. Yeah.
Okay.
Okay.
And
That
so
makes sense.
as a very concrete thing, if there was is there a way to do this in a slice model?
I
Like,
think John Song is the perfect person to answer that question.
like, can you get sequences in a slice model where you could show a sequence of activity and then in calcium imaging, or not calcium imaging, let's say something faster, voltage imaging, and then take that slab, do connectomics on it, and show the connectivity.
But in slice, there's no behavior, right? There's no memory. So what do we want to decode? Did you say slice, burn slice, right?
Yes.
Yeah. So in a slice, we can't correlate it back to a real observed behavior.
Right.
Yeah.
But you could get,
personally, I'm less
the
interested
reactivation of a sequence in a slice, probably.
It's like if we know that this was a slice from a living animal, and that those sequences were presumably, at one point in this review paper, and I know Buzaki has said this a lot, they were saying that these sequences might just exist beforehand, and then they get
that's that whole developmental part of the paper.
okay. But anyway, in any case, I'm not trying to go into great detail. I'm just saying that, I feel like
this actually gets down to, no, this is very relevant, because this gets right into what we mean by a meaningful engram. Does it mean that you've shown that there is an engram, as in there are cells that are tightly connected and that will fire together, or and a sequence of them, or does it mean that we know what they mean, as in we can point back to, like in the songbird, right? Point back to different pitch neurons or something.
Mm-hmm. And I think that defined decoding is a really important point, right? Like decoding can simply mean, based on those neurons, you can say something about the behavior, right? That's what I do in my lab. We show, like by looking at tens of the neurons in the hippocampus in human, we can decode, like with reasonable accuracy, what kind of image the patients are trying to remember, right? But that doesn't mean that information is encoded in that 20, 30 neurons. And it doesn't mean that's the way, right? Now, although in the paper I claim, right, it supports the category neuron, right? The memory is categorized, right, in these kind of different categories to facilitate memory of objects. But still, it's not conclusive in terms of showing that is a code, right? For that is a way of the brain to remember different things, right? So,
well, for our challenge, it's very particular. Decoding means can you go from the structural synaptic connections to function?
Yeah, and when you talk about structural, that by definition means something static, right?
Yeah.
So
wait, Ken, did you just say from the structural connections to function, or did you mean from structural connections to classifying something as belonging to a particular memory or another memory?
in theory, right, if you have all the structural information, right, including something can be interpreted as synaptic waves, right, and even the nonlinear dynamical behavior of single neurons, then right, from structure you can recover all the functional properties
In theory,
right?
Yeah.
But in reality,
The reason I ask is because the memory decoding challenge was supposed to be slightly easier than the brain emulation challenge, as in this functional part is the brain emulation challenge.
The idea is that you could make some very significant assumptions about the dynamics in order to bootstrap yourself to saying this set of connectivity is explaining this pattern of activity.
Right.
Yeah, without so I can imagine that there might be some very fancy electrophysiology models of the neurons that are needed to explain why the phase progression on the theta cycle is happening and stuff like that, and why a sharp ripple will do such and such. And I can imagine just that is essentially assumed by whatever model, and the prediction is these synapses will give this pattern.
Yeah, I'm trying to think about what you just said in the context specifically of recurrent fibers in CA3 and the difference, say, for example, between the recurrent fiber strength or synaptic strength of cells that belong to the same pattern that would fire at one phase of theta and the connections or the connection strength to other cells that would fire on the next one or the synapses from the previous one. And you'd think that even if you just assume that there is an underlying dynamic model of how these cause cued retrieval of an entire pattern and then retrieval of the next pattern because you have, say, a sequence of two previous patterns that then cause the third one to come up. If you have that theory behind it, so the dynamics of it, you can apply that by looking at, say, different strengths of synapses on those recurrent. Very strong synapses would belong to ensembles that fire together, and slightly weaker ones. This could almost be that, that model of bimodal model of just very strong synapses, weak synapses, and basically no synapses. That you'd have, if you have those three, then you could identify what fires together, what fires in a sequence, without having to model the whole thing as at that point you might as well just model it as a function. It doesn't seem that much different or much harder, really.
Yeah. Well, not only that, but as part of the brain emulation challenge, I would suggest it's a suggestion. Take it for what it is. Providing connectivity matrices that are unordered and saying order these, order the cells such that you can see sequences.
Sure. Is that a very difficult thing to do in your mind, or is that a very easy thing to do?
I don't know. I've been asking people, how difficult that is with noise. That one paper by Sebastian Sung that showed a reordering in one example is pretty much where I left off. I'm sure that there is tons of, especially in the hippocampus literature, which reorder cells all the time from a functional perspective. So from a connectivity perspective, there's a whole mirror algorithm, I assume, for reordering things.
Yeah, simply reordering within the graph is a little bit different than reordering based on ephys, but
yeah,
I don't know if it's that much harder, really.
Yeah, I mean, I think it probably comes down to some if it's very straightforward, the number of connections that you have, and the very little noise and stuff like that. I assume it's pretty straightforward. As you get into a real life experiment, it may become very difficult, and simple algorithms don't work anymore. And these are the types of things where you can generate fake data and the answer, and you can push people to develop the types of algorithms that will be able to handle realistic data.
Yeah, I suspect since you're dealing with sparse
Randal,
coding,
sorry, I got to go.
that oh, sure. Yeah, thank you for being here, Dongsong.
Yeah, okay, yeah. Bye.
All right, bye-bye. I suspect that because it's a sparse coding, that doing the reordering to find the auto-associative, so the n-grams that make up the model, memory, isn't going to be super hard, but that maybe in the sequences, so that heteroassociative part, that if you have a lot of overlapping sequences that use many of the same place cells, but in different configurations, that could be trickier. Yeah, that'd be interesting to see how hard that is.
Yeah,
just based on the graph.
We should probably call it. It's
yep.
It's definitely
you're right. You're right.
Journal Club. But thank you so much.
Yeah, my pleasure. I'd love to dive into it more now that some of the work has been done, because I have this separate document with a whole bunch of papers listed and stuff with their abstracts, and so I know kind of where to look now. It makes it a bit easier. Do you think that this process, though, is the right one? We basically say, let's pick CA3, and then we'll just define the next bits, or do we need to do more review? What's your take on that?
So, well, I still don't. I don't have a take on this because I don't know the range of models that are legitimate for the hippocampus. So I almost feel like a hippocampus model, computational model overview might be a good place to go. I don't know, maybe not.
But maybe with a focus on, say, CA3, CA1, and not beyond that, and just kind of focus on that, because there's more consensus around that. You'll find.
Okay. I mean, that's the reason why I'm hesitating, because I don't know. I don't know. I mean, it's like, sometimes I've gotten the feel that I've oversimplified my ideas of what the hippocampus are so much, because there's so much electrophysiology and so much data cycle.
I know what you mean. All these different modes and different requirements for different purposes. It's just a whole bunch of stuff, and then there's so many theories about how it can work. So you get into a big mess, and you could probably tell. But if we're really picking on just CA3, for instance, or CA3, CA1, and we're thinking about this just in terms of getting replay, or how do you identify something that would replay in a certain way, then we have a much smaller set of different models to look at. I think this is way more doable.
My gut instinct is that would be great. Although you're the expert, so this was one of the reasons why I really like the Salmon's paper, because it was connectivity, but it had a very, 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 very straightforward model, and And I was kind of like, okay, now we're talking. Now something that I can even wrap my brain around.
Yeah, that's exactly what we need here. Simplify. Pick something, a small area. Yeah, small bit of the problem. That's right.
And I think this is lost in some cases. If the hippocampus literature is so vast and it's so old at this point, right? That it's almost like, are we not even bothering to talk about these simple models anymore because people are, on the second order or third order tweaks on things, and I feel like I feel like the electrophysiologist might have moved on from the simple models, and the real action will be going back 30 years, 40 years, and saying, oh, now that we have the connectomics tools, we can actually start to evaluate some of these models that are literally 40 years old.
Yep, that sounds likely to me. Yep. Mm-hmm.
Right. Cool.
Okay. Are we still doing a meeting on Thursday, or are we picking a new time for that? Something like that for?
I'm up for anything. So.
Okay. Yeah, I'll just suggest something then, because I guess we should have a post discussion of what papers come up next. And I'm happy to focus on just the CA3 problem. Yeah.
Awesome.
All right. Cool.
Thanks, Randal.
Yep. And thanks, Richard, for recording.
Bye.